Okay. Okay. So welcome back to the sixth and the final online session of the NGS OmicsLogic training program. And today again, I would be talking about your progress. So Dr. Harpreet, if I may share my screen, thank you so much. And it is wonderful to see the progress that everybody has made in this few weeks of the program. And you can see that how much progress everybody as this group is making here. I can see that the server usage has gone up, and also people are using the Code OmicsLogic platform as well as the educational platform. And now to highlight some of the top profiles, of course, we have Aditayo, very active in this entire program, and congratulations again on 11,251 points. And I also see that there were a lot of questions from Aditayo all on the forum, which we will be addressing. And moving on, we also have Ayorinde. So congratulations also to Ayorinde on 7,000 points. And you still have the opportunity to go to 10,000 because you will have access to these resources till November 5th. So you have the time. So take your time to do the courses and the resources. As you know that anybody who has over 10,000 points receives the certificate of excellence in our OmicsLogic programs. So you're very close, Arunde. Keep it up. And we also have Hastened. Hastened, you also need to do the different courses, and this also goes for everyone else. So if you have not yet started exploring the courses or have not been able to follow the online sessions, everything is posted on the forum page. Each of these sessions are posted right here. So let me again take everyone back to the session recordings. So under session recordings, you will be able to access all of these courses, and the links are also mentioned in all the emails that I sent to you. And I'll again send one final email later on, which will have all of the links and all of the resources mentioned so that you can access them easily. So you see all of these resources are here. I mean, the videos of the sessions. So please refer to them, take your time and learn from these resources, from the courses. And without much ado, I would now like to pass it over to Dr. Harpreet. Dr. Harpreet, over to you.
Thank you, Deepsa. So, hello everyone. I welcome you all to the sixth and the final session of this NGS OmicsLogic program. So, in today's session, we will be going to talk about bioinformatics research projects proposals. So in this final session, we will talk about how you can design a bioinformatics-based research project using NGS data and exploring the T-Bioinfo platform. So before heading into today's session, let's review what we have learned during this 1 month program so far. So we started with RNA-seq data analysis, where we learned about various steps that are required for RNA-seq data analysis. Then we also learned about different algorithms that are employed to perform this RNA-seq data analysis, where we have started with the raw reads and finally be able to produce a gene expression table. Next, we demonstrated to you how we can run a pipeline on T-Bioinfo platform to produce a gene expression table from raw read sequencing data. Then we understand about various aspects of this gene expression table. like what are row representing and what are table column representing. So like here, in the columns there are the samples, while in the row there are the genes. Then we understand what are different these ensemble IDs representing. Like ENSG, it representing the human genome. Then ENST is representing human transcript in the ensemble. And then MSSG, it representing the mouse gene. And then we also let you know what is this X-loc representing, that if gene is unidentified in the ensemble, then it automatically get this X-loc IDs. Then we learned about the various normalization techniques, and we learned what this box plot representing, like this box plot representing the different statistics about our data, like upper quartile, median, lower quartile, then what is its minimum value and the maximum value, and what are the outliers. Then we get an overview of various normalization techniques like log scale transformation, and then quantile normalization. And finally, we also understand how this normalization of a data impact the overall results. Next, we understand about the T statistics, where we have tried to understand how we can apply t-test on a gene expression data table using an Excel file. Then we understand the concept of principal component analysis, and we have also tried to run this PCA pipeline to check how we can visualize our expression data table with the using this PCA components. Finally, we also understand how we can visualize our data using scatter plot based on the PCA components. So in the last session, we have learned about how we can perform differential gene expression analysis using the read count data from the RNA expression. Then we also learn about factor regression analysis and why we needed to perform factor regression analysis. At last, we get an overview of various types of machine learning techniques like unsupervised and the supervised machine learning. So now let me ask few questions that what you have learned so far. So post your answers in the chat box. So first of all, which of following are the two algorithms that can both be used before mapping? A, PCR clean tremomatic, B, clean primer and isolase, and then eco and the HR, D, Botai 2 on the PCR clean. Let me see your answers. Kingston, Robert, Gudek saying A. Yeah, you all are right. It's PCR clean and tremomatic. So the next question, which type of file used as the input for the RNA-seq pipeline? FASTA or the FASTQ, which include DNA sequences? Option A, FASTA and FASTQ, that include DNA sequences. B, it is RSCM or text file, which include DNA sequence. Then the C, SVL files or the URL of the files, which include RNA-seq. And D, Excel files. I think most of you have already answered. That is option A. Yes, you all are right. So the next question. So in general, researchers don't use t-test in RNA-seq research. So which of the following reasons it could be? So t-test can be applied only on data that having normal distribution. Then t-test can be applied on RNA-seq data that have read count values. Then t-tests are not suitable for hypothesis-free research, a common approach in which, just give me a second, whole transcript of RNA-seq data. And last, t-test can only be used for categorical data. So again, I think you all are correct that t-test can be applied only at data that have a normal distribution. So the next question, you have run a t-test on a 4 genes in Excel using this particular formula, and then in the column W consider to see if their expression is significantly different in different breast cancer subtype. Which of following genes have significant differential expression? A gene involved in the cell proliferation with the p-value 0.4, and then a gene is involved in the metastasis with p-value 4.0. Then C, a gene involved in a tumor promotion with a value of 0.44. And D, a gene involved in menstruation regulation with a value of 0.04. Sorry. So R&D and editor is saying D. Anyone else? Anyone else? Aditoy and R&D saying D. So both of you are correct. Sorry, this is just a by mistake. It is the option is D. As we know that only those genes considered as significant differentially expressed where p value is less than 0.05. This is just a by mistake. So then the next question, which of following plot provides summary statistics of a data? A, scatter plot, B, bar plot, C, box plot, and D, t-test can only be used for category. Yes, you are correct. This is the option is C, that is box plot, that provide the summary statistics of any data. So we in the last session we have asked you to complete these 2 courses. So how many of you able to complete these courses? So either 1, either 2. Just write 1 or 2. Sorry. Okay. So okay, have you completed the transcriptomics 2? So okay, so now on you can complete this transcriptomics 3. So that's great. At least you able to complete this transcriptomics 2. Now let's move ahead. So let's moving into today's sessions. So today we are going to learn how we can design a research project. So whenever you start any research, so it start with a research question. So what are the major steps that follow to design a research project? Let's look at that. So it's always start with the question, that is you must frame your question or a research problem. Once you started with your question, then you review the literature that is already available and associated with your research problem. Once you're able to go through the literature that is available in different public repositories, then the collect raw data that is needed to address your research problem. And then you just explore and analyze your data and pre-process it for analysis. Finally, perform in-depth analysis to derive a biological significance from the data. At last, once you have performed various types of analysis, just summarize your results that you have obtained from all these data analysis, and summarize in the form of a research article or a research report. Finally, you can communicate your results in form of the research publications. So where now let's look where you can find various types of big data and associated studies, and how it continuously this big data is growing. So let's move to one of the major resource that is for available for the research publications. So can anybody tell me that which resource has the information related to the research publications? So can anybody tell me at which resource you can find publications related to the research? Robert saying PubMed. Anyone else? NCBI. So Robert, you are correct. Both of you are correct. And PubMed is part of NCBI. So let's go to this NCBI. So first of all, we want to, if we want to do any, we want to ask any research question. For example, here I just want to see the publications or the data that is available for the liver cancer. So depending on your research interest, you can search this NCBI resource. Let me just start from starting. So here you just liver cancer or whatever the research interest that you have. So you must search it under the all the databases. So what it will search your query and your research query in this all of these databases. Now let's look at what we will get. So once we search regarding our query, so like I have searched for the liver cancer. Here I have got the information at different repositories, like it is giving me information. There are nearly 2 lakh articles at the PubMed, and that PubMed Central, there are so many articles. And then at the Geo, so if the information related to the gene, that is, there are nearly 44,000 data sets available related to my query. And then there are various other options like geoprofiles, genes. Then about the proteins, it giving information from the conserved domain, identical protein group. So apart from that, at the genome, you will here under the genome, you will get information from the assembly, from the genome, nucleotide sequences, SRA, and bioproject. So like here. So then under the clinical information, here we are getting the different kind of options like clinical trial. So there are the nearly 3,600 entries that belongs to clinical trial associated with the liver cancer. Then ClinVar, it is uh, it containing the information related to the mutations and that are associated with the clinical data of liver cancer. So these kind of information you can find various resources. That's simple. Why if you search on this directly at NCBI. So now let's go over to the PubMed. So at PubMed, so with my liver cancer query, I have find I'm getting nearly 2 lakh results, nearly more than nearly 3 lakh results. So then I can even look into how it started. Like it started with the research, it started with nearly this 1800 something. So it around this 2003 or this at this bar plot that is present in the left side. It giving me information how with every year these publications start accumulating. Like now around 2018 there are the nearly 14,000 publications. So this is how we get different kind of information at this regarding the research articles that are available at the PubMed regarding my research query. So apart from that, you can just go to the advanced option. So if you go to this advanced option, so here you can write your query with multiple query options. Like here, I just want to search liver cancer, and there is one another drug that is associated with the liver cancer. It also asking me whether I just want to search all the fields or whether I want to search under specific fields. Like I can select it and title and abstract. So here I just putting my query liver cancer. So I can add another query, that is, sorry. So I just want to add another query that is related with the very well known drug that is associated with the liver cancer, that is sorfanib. So this is depending on the query that you want to, if you want to design multiple query. So it will search with the liver cancer as well as the sorfanib. So it will give me all the articles, their information related to the both of these keywords, that is liver cancer as well as sorfan is available in my either my title, either in my abstract. So as here we able to sort out these articles, nearly 3,000 articles. So now looking at these articles, we can get what kind of information, what kind of articles we are interested. Even before that, we can review if we want to sort out the review. So I can select it, that if I don't want to select, so it will give me 731 review articles. So how this is how reviews are usually contain all the background of the information. So we, when we want to proceed for any research article, first we always go through the review related to that our problem. So always go through the recent review that is related with your research query or research problem. So here you find, you will get like nearly 700 reviews are available that are associated with the liver cancer. So you can go through the recent one. And apart from, if you are interested only the clinical trial data or meta-analysis, so all these kind of things you can select, and based on even the, you want to see the publication from the recent 10 years or 5 years or the last 1 year. So you can go through that research articles that are associated with your research problem. So once you're able to do that, now let me take you to the another resource. Once you go through these publications, so when I was searching all these publications, I just came across one very interesting publication like this one. So when I was searching these queries, so I just find an interesting article that is associated with the RNA M methylation that regulates sorafenib resistance in liver cancer through this particular pathway. So this particular article is actually just published in this June 2020. So what the authors have done, so here Lin et al. Have actually identified they identified a methyl transferase is significantly downregulated in human sorafenib resistant HCC, that is hepatocellular carcinoma. Further, they observed this depletion of this particular gene, that is MET-L3. Under the hypoxia, it promotes sorafenib resistance and expression of angiogenesis genes in the cultured HCC cells, and it activates autophagy associated pathways. So here further they identified this particular gene, that is FOXO3 is a key downstream target of this particular MET-L3. So finally, but they've observed that the analysis of their clinical samples confirmed that MET-L3 and FOXO3 significantly enhance sorfanim resistance of the HCC by abolishing the identified METL3 mediated FOX3 mRNA stabilization and overexpression of this particular FOXO3 finally restore the sorfanim sensitivity. So overall, their studies, but they concluded that MTL3 has a very critical function specifically to resistant against this particular sorfanim treatment. So this is how you can get the information related to this publication. So now let's go to the another. Once you understand some information and you got the some research article, and if you find in find that this article is very interesting, so you know next thing that you want to do is you want to search the research. Once you go through all those publications, next you will go through the repositories where the data is available to our research problems. So for instance, as we have already observed, there are the data repositories. What are the name? Can you name any repositories where the genomic data is available? I think we have discussed that in our first session. Can you name any repository where which contain the data associated with NGS data? Can you name any repository which have NGS data? SRA, Kingston, okay, that's you are right. Anyone else? Okay. So NCBI is journal. So can you name a particular repository? NCBI is a over mega resource which contain various repositories. So let me just go to the one of the major repository. Apart from the SRA always contain a raw read sequencing data. So other repositories, gene expression omnibus, that is GEO. So if you go through this GEO, this is the mega repository which contain information related to NGS data. So if we go to this repository, so here you just search for again our term like liver cancer was my query. So if I search this, and beside I can also directly redirect it to this particular repository from here as well. So if I click on this, so then I will direct it to the GEO. So here you will find various kind of data. So it's based on your query. So here, first of all, you can select your organism that is given on the right side. You can select whatever the organism that you are interested to work on, like just select on human. So then you can search the study type that you are interested. So if you go to this customize option, so here you can select. So every one of you able to reach to this geo site. Just let me know so that we can together we just go to how we can extract data from a data repository. So just go to this geo and just follow me and just let me know whether you are able to reach to this site geo. Okay, so if anyone else apart from Editoio, so just reach to this repository that is geo. So as I have already told you, search here under the if at the first just put your query here and search it. Once you search it, here you will find various options, like here you nearly 44,000 query you will find. Then you can select a particular organism. So once you select this organism, it will make your query based on this. Like here we can look at liver cancer and the Homo sapiens. So then this is under the study type. So here you can select what kind of data that you are interested. So like here I have selected only the expression profiling by high throughput sequencing. So here I have got nearly 109 data sets for gene expression profiling data. So the where profiling is done by high throughput sequencing. Apart from that, I can select, I have already selected these queries. Like here I can select a strain, so and tissue, and you can customize this as well. And from here as well, entry type, data set, you can select the series and sample. So let me just tell you what is a series. Series is contain the multiple number of the samples that are collected during a particular study design. So it contain more than uh, as many samples that are particularly performed during and study. So then there is even a super series you will find where these multiple series are combined for any kind of analysis. So apart from that, once you get that, here, this is if you look at the top of this interface, here you will get this option, sort by the number of the samples. So here you can select based on whether you want to select your data sets, that first of you want to sort it, like you want higher number of the samples, that is probably our choice. So once you do that, then you will get various kind of data samples here. So various kind of studies. So here I am interested. So I am interested in this, but so at the 17th position, I don't know what kind of study. So here I am getting the same study that I have searched on my, when I have searched PubMed. So then I find the same study here, data set from that study here as well. So if I click on this particular query, that is, if I give you the GSC ID, that would be, I think, help you. I will just paste its GSC ID. You can search it with even this as well. So this ID is specific for every single series. So I'm just interested with this particular data, as this is associated with a study that I have just go through, that where we have find that studies done on HCC samples, and samples that are treated with the sorfanib. Sorfanib, as I have already told you, and HCC cancer-based drug, anti-cancer drug that is designed specifically for the liver cancer. So here I am interested in this data. So even with this GSC ID, for sometimes in the publication GSC, most of time in the publication, the data associated with is submitted in this SRA or GEO. So you can take this ID. You can also search with this ID. Like I just want to search with this ID. So if you search just this ID, so it will take you directly take you to this study. So after we just go through this particular study, and here we will find, at here we will get the details regarding this particular data set, like what was the study here. So it was, first of all, it give you the status that when it was submitted. So like it was just submitted in the April 2020, and then you will get the publication that were also associated. Title of most of time is same, like here it indicate that this RNA M6 methylation regulate sorafenib resistance in liver cancer through FOX3 mediated autophagy. So it also indicated it is RNA-seq data. Then if you go through this experiment type, it will give you the information like this particular study design is based on the high throughput sequencing. Then in the summary, you will get the information related to this particular study. Apart from, and then in overall designs, they usually explain how many samples they have used, like here they, or what kind of sample they have used. So you go through that, like we examine differentially expressed genes between SCC and sorafenib resistant SCC tissues from patients via transcriptomic sequencing. So you will get this kind of information related to this. Then they have Geo is also provided you the publication PMID. So what kind of the publication from which this data was taken. Then you will get here the information related to the samples. So like here we have the 6 samples. Here we have 3 samples that are associated with the normal and 3 samples that are sorafenib resistant SCC patient samples. So then apart from that, you will get here you will can also direct it to this bio project and SRA. We will do that bit later. Now just look at what kind of data is available here. Like here, especially the series matrix file. So this file is usually contain the information related to the samples, all these samples, and in this series matrix file, usually the clinical information is also available. So usually it is processed data, that is the particular authors from this study submitted their input data already which they have processed. Then there is a supplementary file. Sometimes in supplementary files they give you some sample of data, or sometimes they give raw read counts. Sometimes they can also provide you with like sequence uh, different samples information. So and then the main raw files are available under the SRA. So let's go to this supplementary file, and just if every one of you able on this page, just let me know so that we can just download this file. So as this data is only nearly 6 or 7 MB, 690 KB, so we can directly download this data. So everyone of you able to download this data? So just let me know about that. So I have already shared GSC ID with you. So just let me know whether you able to download this data or you able to reach this page. So it means as no one is replying, so you are not able to download this data. So here you will get this. We are downloading only the supplementary file here first. So you can download it by HTTP option. Norman, yes. So as this is the zip file, gz, so just unzip that. If you able to unzip that file, and just let me show you what kind of file you will get. So if you don't know how to unzip a file, you can just Google it, how you can unzip any .gz file. So here we have got a data like, so let me just paste it to into an Excel and let's see what kind of data it is. Before that, just look at, we can even look at here. This data, this particular series patient, patient and normal SCC and sorafenib resistant SCC genes, FPKM values. So they are also giving that here this particular data contain FPKM value. So if we copy that and just paste it here, paste in Excel. So just look at what kind of information that we have got here. So it giving us an ID. So I think gene ID is something is written on that. I just changed that into the ID. So as our most of resources are taking ID, so let's see how we can analyze this data on our T-Bioinfo platform, or based on the information already that based on the different algorithms or different methods that are available at our T-Bioinfo platform. So here, if we look at this data, in the first you will find gene ID. So this ID, so then it's a gene name. Then it also giving us the locus, and then in the it is only for the two samples. Here for, these two samples are given. One is for the normal, other one is sorfanib. So as we don't need all this information for any kind of analysis, we can take, I just copying and paste again. I'm just pasting this information here again, and oh, sorry. So here, as either I can take a gene ID, either can I take a gene name. So let me just delete these two columns. I'm deleting this. So now we have ID, and then we have FPKM values for the normal samples, and then we have a FPKM values for the other sample, that is sorfanib resistant. So now we can do, as I have already shown you, we can first of all, once we get the data in form of FPKM, so whether we can apply t-test or not. Let me just ask one question, whether we can apply, as this table has FPKM value, whether we can apply t-test or not. Can anybody tell me? Yes, we can. Kingston saying yes. Anyone else, whether we can apply t-test on it or not? Arundi saying yes. Yeah. So since it is FPKM values which is normalized, so we can apply t-test here. So one of the major tools that actually we can use, that is also, if you want to do your t-test in R, then you can apply other packages as well, like uh, let me just, I got it out of my mind, the name of that method, Wilcoxon test and Welch t-test, and we can also apply t-test in Excel. So before the t-test, let's look at this the values, so that we can understand whether we, as we have already told you, what kind of analysis we can perform. So first of all, as we have already told you that we cannot, we usually don't directly proceed with our data. So first of all, we convert them into the log transformation value, and we apply a normalization, that is log transformation. So as I have already shown you in our previous session, so let's do it today again. So as this data, so how we can apply the log values? Let's do it here. Just put equal, then do ln. So then we have to select the value on which we want to do, we want to transform log scale. And then as this is 0, so what we should do? Anybody remember that? Whether we can directly compute log normalization, can apply directly log normalization on 0 values or not, or what we should do here? Anybody remember that? Yeah, plus add, we have to add +1. So just add +1. So we can drag it. We can drag it all the columns. So before even applying the log normalization, always take your our values range. So either we whether we need to even apply log normalization or not. So I think in this case we don't need it even. I think it's already log transformed values. Okay, no. As there is large variance again, if we observe here, this is 174, 195. So based on this analysis that we need to transform these value to log normalization. So again, I just copy this. So this is normal or control. This is sorfanib resistant. So there could be multiple sample, as here we are just taking proceeding with the only the two samples. One is belong to the normal or the control, other one is sorfanib. Although in the data there are the 6 samples, you can perform any kind of analysis. I'm just showing you just with this particular analysis. So now as we have this log transformation values, so I can just so now we can apply t-test on this. So anybody remember how to do t-test? So before even the t-test, we can also filter out those genes where there is zero values, all the zero values for every sample. We can filter out that. But here I'm not just doing that, but we should filter out those genes where there is zero values. So you can always delete all those genes where there is value, zero value, like this, both the samples. So there is zero value. So we need not to proceed with these kind of genes. So I am just here doing only the, I'm taking this and I'm not removing here. So let's apply t-test. So it will take array. Here as we have only two kind of samples, so we taking array one, this is normal. So then we have to select the array 2, and then as we want to see whether it is upregulated and that downregulated, then we decide this 2. Then type of test that here we are using 2 pair, so the 3. So there is something wrong in the formula what I have done. I just do it again. Okay, it cannot compute the standard deviation as there are only the two samples. I just copy this sample here as well, just to paste values. So I'm just considering these two as two different samples. So as t-test need to compute standard deviation. So if we have only two samples, then it cannot compute the standard deviation become automatically 0. So that's why that t-test cannot be applied on the two samples. It was showing the error. So let me just consider this as one value. Let me try again. So this is something I'm just getting some problem here. Let me, just let me try it somewhere else. Here I have multiple samples. Okay, sorry, I just not selecting the array. So even I need not to do, I can delete these columns. I know what the problem I'm doing. Okay, so here I'm not selecting all the samples. Like here I have to select t-test. I just selecting a wrong array. So array is all these samples. No, no. It should be applied. So this is, I'm just facing some problem. Just I will see it later. So simple, it is always with the formula. So I just try this here. You can take this. Let me just do it. So the problem was that we are, now I can understand. So so the main problem here, as we have only the two samples, so we must have multiple sample to apply t-test, at least more than two samples. Like here we have in class 1 we have only zero one sample in other class. So if we have more than two samples, we can easily apply t-test. So like here I have multiple samples. So I just take one class. This we just have to apply the t-test. Like here we can directly, then we this compute our p-value. So this is how we can perform. So here, as I have already told you, so let me just, then we can add a filter here. Okay. So then based on the filter, then we can sort our genes. So I need to paste even the gene, insert the column, gene name. So then based on this, we need to apply a filter again. So then based on the p-value, we can sort it. So smallest the p-value is, the more the significant. So here the gene I have selected are not a significant differentially expressed in these two classes, because these are actually to belong to the same class. So this is how we can perform simple t-test on this particular table. So beside, once you get the table, but kind of analysis that you can perform. So then you can also perform the PCA. So first of all, if you once you got your data and you just log transform, so even if you don't log transform, you just quantile normalization. In under quantile normalization, it give you an option like how you can perform your quantile normalization, then PCA. Now let's go over there and let's see how we can perform quantile normalization if you have gene expression data table. So if we go on our resource, so under the utility, so here you need to provide your file. Okay, so let me just make this file as a, so when I have this data, I can just paste this data in a text file, in a new text file. So like here I have just create a new file. So we have to remove the last line and we need to provide ID as it is given, then the sample 1, sample 2. We could have a multiple sample. Let's just save this file. So it's as input file. Now let's go over platform. So under the first, if you go over the platform, under the utility, so under this particular option, data integration and modeling, here you find sub option data mining, and you can apply various kind of analysis. Like here, once we have our data, then we can just upload our file. As I have just saved this file, this input. So next we can proceed. Please just waiting. Okay, once it's done. So next what we can do, it's asking whether we want to perform quantile normalization. So let's go for the quantile normalization. So it's asking, as you have FPKM values, so if you have not just transformed these value initially in the log normalization, you can also do this at our T-Binfo platform. Just transform table to the log normalization, yes. Then log normalization value, then we can select a threshold that is for quantile normalization. Uh, Here I'm selecting a 10. So once you have run this, then you can also run the PCA. So here we are not using any filter. So then it's asking whether you want to transform even here. So as we have already transformed this table into the log normalization, so then you just select the SPD options, and then standard, you want to perform standardized table rows before PCA, you want to do yes. Then table column, you want to do that, we are doing yes. So once we do this, then we can perform log normalization. PCA and QL. So you can run this pipeline. So while this pipeline is running, let me show you how we can upload our data. Then the next, so this is like already processed data in form of FPKM values, but here we have only samples for the two samples, data for the two samples. So it could be as authors just provided our us this data. So Then we can also proceed for our own analysis, that we want to do this on SRA. So we want to extract the SRA table. Now let me demonstrate how we can actually download SRA table. And the SRA table is contain the information related to the raw sequencing data. So let's go to this particular study. Related to this particular study, let's go to the SRA. So once we are on SRA, here we have again find the 6 samples. For here, what kind of samples that we are getting? Like here we have the first sample, that is 5.9 GB. So then another is nearly 8.2 GB, 6.7 GB. So these are the raw deep sequencing files related to these 6 samples. So how we cannot just obtain this data like directly, because this is like, even with the 6 samples we are like 50, 60 or 70 GB data. So it's impossible to design any research problem like that. So what we will do, let's just go to, every one of you are on this page, just let me know, so that we can understand how we can extract this data and directly upload on our T-Bioinfo platform for any kind of analysis. Just write yes if you are on this page. So this is how we, okay, just let me know if you are on this page. If you're able to reach to this SRA, let me just demonstrate it again. So once we have selected this particular GSC data, here once you go to this data, at the bottom side, here you will find the relations like bioproject and SRA. So at the SRA, here this will just direct us to the directly to the SRA, that is the samples that are associated with this study that are available at SRA. So once we just click on this SRA ID, then we just move to this page. Once we are on this page, so then we can directly just go to the send. Here you just select the first option, that is file, then the format it asking. So the summary, or then just click on this drop down. Here you will find 4 options: summary, run info, accession list, full XML. So here we want to get the information related to the run info. So if you do this, so every one of you able to just reach to this site and able to download this data. This is like, here we are downloading SRA data. So we are not directly downloading whole data. We are just downloading the information related to that. So that we can directly upload on T-Bioinfo platform. So if any one of you able to download this SRA table, just write your answer in the chat. Okay, so if you're able to do this, now let's go to this SRA table. Just let's open it first and let's see what we got from this SRA table, SRA run table. So this is SRA run table. So here what kind of information that we are getting. This is giving us a run ID, then the information related to the release date, loading data date, so all the bases, this kind of information. So then beside it also giving information related to, like here RNA-seq data, then these are cDNA, then this is the paired samples. Even this information is also available if you look at this platform. So from here even you come to know what kind of data it is, whether it is single end data or whether it is pair end data. So even at the library layout, you will get this information is pair end, and this information is also available at our SRA table. So this kind of information table, once we just get able to get this table, so every one of you able to got this table. So if you able to do this, so then we just see how we can upload this data on a T-Info platform and to perform any kind of analysis. Just let me know that every one of you able to download this data. Oro saying sorry, poor network. Just let me again demonstrate it. Meanwhile, if you face any other problem, once you are on this SRA page where you find there are six samples, and on the six samples here we will find an option that is given at this right side. So here it is send to. So if you click on this drop down, just go to the file and then under the format select this second option, that is run info. If you able to do this, then just create file. Once you create this file, you able to download this table. So once we have this table, now how we can run RNA-seq data analysis. So I will go to as my this pipeline, and just let me just before that, let me just show you how we can directly upload this SRA table. So let's go here. As we here, first of all, what kind of analysis that you want to perform. Accordingly, you can select your choice. So here I am just want to perform RNA transcriptome, RNA-seq analysis. So just let me go to this. So first, once you are on this page, just let me before that, just let me know how many of you able to download that SRA run table. Any one of you able to download SRA run table? Okay, Aditoy, that's great. Robert, that's great. So once you able to download that page, that SRA run table, then just go to this area of analysis. That's great. Every one of you able to do this, I think. Okay. Once you download it, go to this area of analysis. And if you want to do our RNA-seq analysis, just go to this first option. So as here we are not running a demo, here we are just directly dealing with the real data that we have obtained from a study. So then just click on RNA-seq. Once you are able to select it, then first of all, it asking you so many options here, like first type of the reference genome that you want. As this is already this genome already we have, so we are using our model genome. Then it is asking the format of NGS data. So can anybody tell me what kind of information or data is available in the SRA? So can anybody tell me what kind of data is on SRA? Whether it's a FASTA, FASTQ, or SAM file. So can anybody tell me in the SRA file FASTQ, editor is saying yes. As SRAs, yeah. SRA is as self as it is its name even indicating that this is sequence read archive. So there is a read data, and read data is always in the FASTQ. So we are selecting the format of NGS FASTQ. Then as we have seen that this was parent data, and the organism here was the human, as these are human samples. So once we able to do that, now let's upload our file. So here you will find various options. As this particular table was SRA run info, so this will be uploaded under the SRA run table upload. So if you click on this, and now you can upload your data, that is SRA run info. That this I have saved this like this, and your table is also downloaded like this, SRA run info. So once you able to do that, now here your data is getting uploaded. Probably it won't take the CSV file. I have to convert into the text. Let me just before that, I just need to copy it into a text file. So we cannot upload CSV file. So let me just convert it into text file. So I just copy that table and then just paste. So I have to remove the blank line from end of the file. So now let's save it into a new text file. So this is SRA run table. So now let's go over here again. I can cancel it as I have just uploaded a CSV file. But on T-Info platform, you cannot upload your CSV file or Excel file. So I just cancel it. Now I am adding this file. Here I am adding this SRA run table, which is in text format. So once you able to do this, now let's go to the next section, that is asking a groups. So here we know that the first three samples, I can add another group. So as I know, the first three samples were my HCC samples or the control sample. The next three samples are actually the samples that are sorfanib resistant. So let me just do the other group. As I just want to perform differential gene expression analysis, so just import these samples into two different groups. I know the first two, you must know what kind of samples they are, and this information, from where you get this information, you can just, if you look at here, so it is showing first three as sorfanib resistant, sorry, and next three, then you can match your IDs as well. So then it's saying the next three samples are SCC. So we have two groups, both belong to different classes. So once we able to do this, now let's move to the contrast. No need cancel, we can cancel it. Now let's start with our pipeline. So once we able to start it, then we can, next step that we can do, PCR clean, then the Trimomatic. So this is pre-processing step that we are going to do, that is PCR to remove the PCR duplicates. Trimomatic is removing adapters. Then we can perform high set, or even we can directly just go tight. Okay. So from this RSCM expression table, I'm just again able to generate the FPKM table. So then if I want to perform differential gene expression analysis, I have a question whether I can perform differential gene expression analysis on this data or not using this DSEC or HR. So can anybody tell me that whether I can perform differential gene expression analysis on this data or not? Yes or no? So if I have selected here, I have selected the count values. In that case, I can perform DSEC. As I have just selected the FPKM values, in this condition, I should not apply this HR or DSEC as it requires read count. So if I have selected, in case of when I'm just doing RSCM expression table, so if I have selected the counts, then I can perform, but here I cannot. So this is depending on the different options that you can explore, and then you can perform various kind of analysis. So here I am just doing RNA-seq for FPKM. So this is how you can perform various kind of analysis on your data. once you able to I mean, this is how you can design your own project and then perform various, explore different options. And like here, even the under the area of analysis, you can, once you have obtained your RNA-seq, then you can perform unsupervised analysis. You can perform various kind of PCA. You can run PCA, and then you can also perform clustering methods like k-mean or the h-clust. So this is how we can proceed with even the raw, how we can upload our data on our T-Bioinfo platform and perform any kind of analysis. So are you people able to complete this pipeline and just able to upload your data? So just let me know whether you able to perform, able to this, yes. Are you able to perform, run this pipeline? So this is how you can do various kind of analysis from the data that from what the different data sets that you have obtained from public repositories to design your own research project. Now let me move to back to the presentation. So you can run PCA, and then you can visualize which are samples, that whether these samples are getting separated or not. So that you can do both by PCA-R library, as well you can also do in Excel, that I have shown you even initially. So let's, then this was the SRN table. So this is to design any research project. Once you able to obtain your data, so then the pre-processing steps are, it started with the pre-processing. Then it align your reads on a reference genome. So then you will perform the mapping. Then you can quantify, you can perform the quantification. That is, you can quantify your gene expression values, and you can generate gene expression value in FPKM, read counts, and TPM. So depending on the your choice of analysis that you want to do, so that you can do on T-Bioinfo platform, and then we can perform the summary statistics. Like we can generate the box plots, and we can generate the bar plots and visualize our data. Then we can also perform normalization. As I have shown you, we can perform log normalization on our data, and quantile normalization. Then accordingly, we can hypothesize our testing. That is, applying t-test, we can understand whether some genes are significantly differentially expressed or not based on p-value. Then we can apply various data mining approaches that I have already shown you, like we can apply PCA, and in our previous sessions, we have already shown you how we can apply the PCA and how we can understand whether these samples are getting differentiated from each other by just visualizing on PCA scatter plot. Then we can also apply various classification, supervised or supervised classification approaches. And finally, we can annotate those genes to understand which are genes that are playing important role for various path in different pathways. In the last sessions, I think already we have shown you DNA differential gene expression analysis, where you can understand which pathways are getting disturbed in different kind of the samples. So this is how the RNA-seq that we have already shown you, that how we can run RNA-seq pipeline. Decide if you are more interested in mutational analysis. As in mutational analysis, even you just need the FASTQ file. So you can even perform the mutational analysis using a mutational pipeline that we have shown you the second session. So let me just briefly let you know how we can do it. So once if you go to this server, go to this area of analysis. So here under the genomics, you will find this pipeline mutational variant parallel analysis. And once you are here, you again upload your file and perform mutational analysis. Like here we have already shown you. First, you will do the PCR cleaning once you uploaded data, as I have shown you today, how we can upload our own data in PCR at our T-Banfo platform. Then you perform PCR cleaning. Then on Batai to do will map your then do go for the mapping where your which allow your reads to align with the reference genome. Then perform the Sturka and snipper that will eventually generate a significant mutational between different kind of a sample conditions. So if you are interested to perform kind of genomic analysis, genomic mutational analysis, so you can perform this analysis on T-Banfo platform very easily once you have your own data. So now I think every one of you before that, just let me know next you have to propose your research proposal. You have to give us your research proposals. So this is okay. So you can choose your own research problem, as well as you can take different research proposal examples from our T-Banfo platform. So let me just copy this link and provide you. So here, either you have to plan your own research project, either you can choose any research projects that are available at this particular platform. So you can decide different research projects from here, select and decide. I think now I should hand over this session to the Deepsa so that she can let you know how you should submit your project submission form. So over to you, Deepsa.
Hi, Harpreet.
Okay.
Thank you for showing this. I will just take over now.
Okay, okay. No, just okay, Mohit sir, it's over to you now.
Thank you for the sessions, live sessions, and these tutorials, Dr. Harpreet. And I would like to ask everyone, how's everyone doing? So we learned a lot in 2 weeks. So how do you find this training so far? So please go ahead and write it down or tell me, like unmute yourself. So one of the objectives is to again complete this training with a research proposal. So I'm trying to gonna give you a overview of how it is and what are the possibilities. So we have just finished the stage one here. So the stage one is where we have learned about these technologies, saw some of the applications, and saw some of the pipelines, how they work, learned about the algorithms and the output, learned to analyze the data. And we learned about two technologies in this short period of time, that is genomics and transcriptomics. And also then today we learned about how do we find the data online and use it directly to the T-Info server, which is like a very I think important integration in terms of doing real-time research. So now there's an opportunity to be able to propose your own research or to learn from examples. So there are two options. So let me share my screen and tell you more about this to you all. Okay, so for the next 15 days, for the next 15 days, we will be depending on this place, the forum and the organization page. And I think you all are pretty much now familiar with this place. So today was the last scheduled session. And here, as you know, that you will find updates from everyone. Also, Ilya is commenting based on some of the problems that you have pointed out. And then we have the forum. In the forum, you will find a project submission form. So this is a dynamic form which you can keep on updating, like putting information in it, and it will get updated. So the purpose is to be able to fill this form, all of you, where you can propose something like a project title, like a problem statement that you think could be worked upon using big data and using transcriptomics and genomics data especially, and which we covered in this program, and the link to the raw data from the repositories that Dr. Harpreet showed you today, and then the reference publication. So if there's a reference publication, it actually helps a lot to devise a strategy. So these are some of the fields that you have to fill, and these are some of the examples that you can learn from how to do this. So these are some of the people who have already done this, and many of the participants from Africa have also done this before. Some of them are working with us as interns. So you will find here that the outcome in the end, when this program ends, we would be able to, if you work through this project, and then we will be able to get you to a place where you can record and do this entire presentation about the project and how you did the analysis and every step. And you will find the problem statement and the research title of the research, and also a video explaining the entire research. So there are few examples here to learn from. So this is already being shared by Dr. Preet. So please go to this link and see it for yourself. And once you have something that you wanted to, put down. So it does not have to be a like a high level, or what you can say like a complete research proposal. You can start with something that you have in your mind, and we can help you refine that idea. But we do need you to submit this whatever idea you have here, so that we can review it, and then we can plan a session next week, or the week after. It depends on the submission. So if you are going to submit, then we will be able to have a review session on this, and we will be able to discuss it with the group. So that's the purpose. And do let us know if you have any other questions and problems. Okay, another thing. So the second aspect, if you don't have a project proposal or a project idea that you want to work on, there are project examples that are there for you to take. So there are two projects that are already available with this course that you can actually take and learn from. So modeling precision medicine and Spodoptera frugiperda. So these are two projects that are available, and there are other projects also that you can request if you have a proposal. So if you read about this project, and if you think that you can do something more on this data set, that is also possible. So working on already published data sets, published stories, and if you want to do some more analysis, some other downstream analysis, some different questions that you wanted to address. So those things are also possible. It's just that we need it in a form of a proposal, which is there in the forum, which you can submit anytime, and you can keep on revising this as well, as it is a dynamic form. So this is the process, and if you have any questions, please go ahead and ask. So the progress has been really amazing. You guys are doing really well and started off really well. And can I work on a project on malaria? Absolutely. You can work on a project on malaria or any other disease. You just have to look for the data set and the publication and those associated fields that you want to write about the research problem especially. So sure, that would be great. And Editio, there is no such ultimatum to the research proposal, but you have the access to the resources for another 15 days, all the resources, the educational license perhaps, and then we can switch to some other basic license where you can take courses. But for this, I think if you do it sooner, the better the chances of getting it peer reviewed, and then think about the next step, if there is a fellowship or scholarship, or how to do this to take this to the next level, if it is worth the project proposal. It depends on the project proposal. Yes. No, Kingston. The proposal has to go through several steps of review. So it's important to have a solid proposal with a good abstract, right? So if you have that hypothesis ready, that itself takes a lot of time because you have to look into the data and you have to look into the feasibility of your if that data is able to will be able to give you answers to your question or not. So Dr. Harpreet here is an expert, and I also try to contribute to help you in those steps, whether if the question is valid or not and stuff like that. So we'll go through that process of refining the proposal and trying to figure out a way strategy. So it will not be a complete project, but it will be surely, I think, depends. Again, it depends on the research question and the analysis that you are planning to do. Okay, if there are no more questions, I would like to ask Adi Kunle, who is a faculty with us and have been participating in all of our sessions and trying to help other students by communicating with them about their problems and trying to solve them, mentoring them. So I would like to invite Adi Kunle to talk about his experience and to the participants who would want to work on project.
Thank you very much, Dr. Mohit. Thank you, everyone. Can you hear me? Hello, can you hear me clearly?
Yes, yes, yes.
Okay, great. Yes, good afternoon, everyone. I'm happy to be speaking to all the participants today. It's been a jolly ride, I would say, for the past 2 weeks, though tasking. I'm happy that many of us have come this far. And I want to believe I have people we can work together also to advance the Africa OmicsLogic program together. So I would like to encourage us all for the next 2 weeks. Let us take our time to do this project and lay our hands on some of the tasks to ensure we get more benefit from this platform. And like Dr. Mohit said, going forward, the projects, ideas that comes up are things we can work together on, okay, guiding the participants. But importantly, I want to say that for many of us, especially for those of us who have had good experiences with this program, I would want to encourage us to please work together to advance further the OmicsLogic Africa program. So as a matter of fact, I'll be reaching out to some of us. I've interacted with some of us via WhatsApp. So after now, in coming days, I'll be reaching out, telling us more specifically on how we can work together, especially for those who are faculties in various universities. I'm based in Lead City University, Nigeria. And of course, we are seeking out ways we can integrate this into the curriculum here, and the management is very much open to it. And for some of us also who have, gone through this program, we have seen, the benefits and the opportunities that lies in this study. So please, I'll be reaching out to us, discuss how we can also extend these programs to our various institutions. It's a program that is meant for institutions across Africa. So, and in fact, there are some collaborations and, grant opportunities that we are currently working on. So it's meant to be an African-wide program. So for those who will be interested, please do not hesitate to let us work together to extend this to other participants, students, both undergraduate and postgraduate level. So that would be my comment for now. And so in case we also need any further support or assistance, I can also be readily reached, just as I know BIPSA has always been available for all of us as the, the community manager. So please, that will be that from me. And also there will be other programs coming up, like Dr. Mohit mentioned the other time, other advanced programs. I believe we are already part of the community now, and we can always, interact and engage and be part of future programs. So thank you, everyone. Thank you, facilitators, Dr. Harpreet, Dr. Mohit, Bipsa, and also Elia. Thank you very much. Over to you, Dr. Mohit.
Thank you so much for your comments and all those scope that we would want you to work on. And we are working with some of the universities to be able to do what we actually just discussed, that Adikunle just discussed. So we are hoping that we'll be able to collaborate with some of you and do this on a larger scale. But within in this program, I think there is one other aspect that I forgot to mention, is that you can collaborate within this program, and maybe you can also ask other people out of this program to collaborate for a project. So those are also possible. So that actually helps to have this in a shorter period of time, more people in doing different kind of analysis together. So that's also a possibility. So again, we will be meeting again. We will be meeting next week, perhaps, or we will schedule that, and you'll receive emails from Deepsha about that, and then we can discuss further about the proposals. And it's been a pleasure being with all of you, seeing your progress and learning, and saw a lot of new insights that from the coding platform, especially, that we did not expect it to go well so far, but it has been really, everyone has been able to understand all of the aspects, and that's really great to see. And I understand that it's going to take you a while. So this 15 days, please use this period to get more familiarized with the entire way of learning, so that it would help you in your research. For today, I would like to thank everyone for joining, and we'll see you maybe next week.
Thank you so much, Dr. Mohit, Dr. Harpreet, and Adikunle. And I want to thank everyone here for attending this program and for your wonderful performance during this time. I will be sharing all the resources soon. Please feel free to reach out to me anytime if you have any questions about the resources, so that I can help you out. And we look forward to seeing your projects and working with you in the next 2 weeks and in the future as well. So thank you, everyone. Yes, okay, sure. Dr. Mohit, can we have a quick photo shoot with everyone, just like a screen share and photo?
Yeah, so I thought we would do that in the next session. So we will invite everyone, and we'll have a session on proposals, and
Yeah,
then we can
right.
do
Yeah.
that.
Okay.
So
Yeah.
this is a heads up for everyone. So next
Yeah.
time
So
we'll
for the
have
next
that.
session, then okay, we will have this. So we'll have a snapshot together, in Skype only. I mean, in Zoom only. So yeah, see you all soon, and I'll let you know about the upcoming session. We'll schedule one in the coming week. Thank you.
Thank you, everyone. Thank you, Deepsa.